What I'd like to do today is to start discussing some more complex microbial communities.
My name's Michael Sadowsky, and I'm a professor in the Department of Soil, Water, and Climate, and I'm the director of the BioTechnology Institute at the University of Minnesota.
The bacteria that usually attach into unformed biofilms are, generally speaking, aquatic by nature.
I'm a bacteriologist by training. My area of research is involved in environmental microbiology, from finding microorganisms out in the environment, determining where they came from, looking at pathogens and their interaction with algae and plants. And I also actually work on the ecology of intestinal bacteria. And more recently, we got involved in this project on the Minnesota-Mississippi Metagenome project. Hey, do you know what's going on with the last set of samples for metagenomic analyses?
Mike Sadowsky is one of the brilliant scientists of our era. I don't know how he manages all that he does. He has multiple projects going. He manages programs. He has graduate students and postdocs and still teaches undergraduates. He is engaged at the national and international level. He keeps traveling to all these great places and won't take us all with him. He goes to France and he goes, to Asia and knows people from all over the world.
Each color on the pie chart represents a different major group of microorganisms.
Mike's incredible. Mike does the work of easily 3 to 5 people at any given time. He can keep track of so many projects at once. Mike's job is basically to oversee and guide all of us through our different projects.
We don't know what the majority of microbes are in fact doing.
He does a lot of taking our results, our data, and giving talks, getting that information out to the public. So Mike's really the public face of what's going on with our teams in the lab.
To understand what I do now, it's really important to see what we used to do as scientists, because things are evolving and changing over time. In the early days, scientists really acted almost by themselves, or maybe interacted with a colleague or two, or a graduate student or a postdoc. But nowadays, science is becoming much larger. Well, sorry we missed the last couple of meetings, but we need to catch up now. And this big science requires teams of individuals with various expertise to understand and in a rapid way get a feeling on what's going on in a particular system. Where do we stand with Itasca?
To be a force in science at his level, he needs to be able to interact with people really well. And so he has to be social, and he's very social.
So my training is in bacteriology and genetics and genomics. But I also have to have team members that are experts in bioinformatics, that is, how do you process large data sets. I work with colleagues that are experts in physiology and diversity of microorganisms and the ecology of microorganisms. So we all work together as a group in a synergistic way to take all this information and process it and understand it and present it in a way that the public and our colleagues can understand it.